The overall goal of this procedure is to screen plants infested with root knot nematodes. First, grow healthy seedlings in sandy soil or growth pouches. Next, produce an inoculum of highly infected second-stage juveniles of root knot nematodes. Then, properly inoculate and infect the seedlings. Finally, evaluate the development of nematode egg masses on the host root system.
We will be demonstrating high and low throughput screens of plants with root knot nematodes. And the main advantages of the high throughput system is the need for limited space for plant growth and ease of evaluation of the infection.
So these approaches will result in reproducible and uniform infection, nematode infection of plant roots, and therefore a better evaluation of the host status of the plant. So generally, individuals new to evaluating plants with root knot nematodes are likely to struggle because of not using the proper developmental stage to infect the roots with nematodes, and also by using several-day-old infective stage juveniles, which typically have low infectivity.
Demonstrating the procedure will be Hagop Atamian, a graduate student in my laboratory.
I will be demonstrating the procedure for screening in the growth pouches.
For pot assays, plant tomato seeds in a single pot filled with organic rich soil, such as sunshine mix. Maintain in a greenhouse at 22 to 28 degrees Celsius. After germination, fertilize once.